Welcome to this lecture on animal biotechnology. Today, we are diving into one of the most fascinating and revolutionary tools in modern genetics, TALEN gene editing. Imagine having a pair of microscopic scissors that can find and fix a single typo in a library of billions of letters. That is exactly what TALENs do inside an animal cell. Let us explore how these precision molecular scissors work. To truly appreciate how TALENs operate, imagine you have a massive library containing billions of letters. This library represents the DNA inside an animal cell. Now, suppose there is a single spelling mistake in one specific book that causes a terrible genetic disease. How do you find and fix that exact mistake without damaging any other books? This is where TALEN gene editing comes in. They act like a pair of highly trained, programmable molecular scissors. Instead of just cutting randomly, these scissors can be programmed to read a very specific sequence of letters and make a precise cut exactly where the mistake is located, tricking the cell into fixing the error. Now that we understand the basic idea, let us look at what these scissors are actually made of. In scientific terms, TALEN stands for transcription activator-like effector nucleases. They are artificially engineered enzymes created by fusing two distinct protein parts together. The first part is a DNA binding domain, which originally comes from a type of bacteria that naturally infects plants. This domain is incredibly unique because scientists can customize it to recognize and bind to any specific sequence of DNA base pairs. The second part is the DNA cutting domain, which acts as the actual blade. Together, they form a highly targeted tool for genetic engineering. Building on the structure we just discussed, let us focus on the cutting mechanism itself. The cutting domain attached to the TALEN is an enzyme called FOC1. You can think of FOC1 as the actual blade that snips the DNA strand. However, there is a brilliant built-in safety feature. A single FOC1 molecule cannot cut DNA on its own. It only works when two of them come together to form a pair. Because of this, TALENs must always be designed in pairs that bind to opposite strands of the DNA. This dual binding requirement ensures that the cut is made with absolute pinpoint accuracy, preventing unwanted damage to the cell. Understanding this pinpoint accuracy helps us see why this technology is absolutely revolutionary for animal biotechnology and medicine. Before tools like TALENs existed, altering a specific gene was incredibly difficult and often resulted in random, off-target mutations that could severely harm the animal. Today, we use TALENs to create genetically modified animals with incredible traits, such as cows that are completely resistant to devastating agricultural diseases. They're also being used to engineer pigs whose organs might one day be safe for human transplantation. Furthermore, they allow scientists to recreate complex human genetic disorders in laboratory mice, which is critical for testing potential cures. With these incredible applications in mind, we must ask a crucial question. How do we guarantee that our molecular scissors only cut exactly where we want them to? To understand why TALENs are so safe and effective, we need to look at the mathematics of DNA sequences. As you know, DNA is made up of 4 chemical bases: adenine, thymine, cytosine, and guanine. If we want to cut only once in a massive genome containing billions of bases, our target sequence must be long enough so that it does not appear anywhere else by pure random chance. To ensure this, we rely on probability. This brings us to the mathematical foundation of target specificity. We can calculate the probability of a specific DNA sequence occurring randomly using a simple exponential formula. In this equation, P represents the probability of finding a specific sequence of DNA by pure random chance. The number 4 in the denominator represents the 4 possible DNA bases that can occupy any given position in the sequence. The variable n represents the total number of bases in our chosen target sequence. As the value of n increases, meaning we design our TALEN to recognize a longer sequence of DNA, the probability P decreases exponentially. This tells us that longer recognition sequences are exponentially more unique. Now that we have our formula, let us see this with a concrete example. Suppose we are designing a single TALEN to edit a specific gene, and we want to know if a sequence of 10 base pairs is a safe length. We need to find the probability of this exact 10-base sequence occurring randomly. We start by taking our probability formula and substituting n equals 10, because our TALEN is designed to recognize a 10-base sequence. Calculating 1 over 4 to the power of 10 gives us a probability of approximately 0.000000953. This means there is roughly a 1 in a million chance for any random 10-base stretch of DNA to match our target exactly. Having found the probability for a single stretch of DNA, we must now see how this plays out across the entire genome. To do this, we use the expected occurrences formula. Here, E represents the expected number of times the sequence will appear in the entire DNA. We find this by taking P, which is the probability we just calculated, and multiplying it by N genome, which represents the total number of base pairs in the animal's genome. If the expected value E is greater than 1, it means our molecular scissors will likely cut the DNA in unintended places. Therefore, we want E to be as close to 0 as possible to ensure the safety of the animal. Let us apply this expected occurrences formula to our previous example. We are editing a mouse genome, which contains roughly 3 billion base pairs. We want to know how many times our 10-base sequence will appear. We take our calculated probability of roughly 1 in a million and multiply it by the genome size of 3 billion. The result is 2,859. This means that a 10-base sequence would appear approximately 2,859 times in the mouse genome by pure random chance. Physically, this tells us that our TALEN would cut the DNA in thousands of unintended places, which would be disastrous and likely fatal for the cell. The alarming result we just calculated perfectly illustrates a fundamental design principle of this technology. A 10-base sequence is simply too short. It appears thousands of times, and cutting at all those unintended sites would destroy the cell. This is exactly why real TALENs are never used alone. They are designed in pairs, a left TALEN and a right TALEN. When they work together, they require a combined recognition sequence of 30 to 40 base pairs. If you plug 30 into our probability formula, the expected occurrences drop to practically 0. This dual recognition system ensures the sequence is absolutely unique, and the cut only happens exactly where we want it. Now that we know why pairs are essential for specificity, we must consider how they physically fit together on the DNA. A functional system requires the left and right TALEN proteins to bind to opposite strands of the DNA. Once bound, the FOC1 cutting domains attached to them must reach across the gap and meet in the middle to form a dimer. This gap between the two binding sites is known as the spacer region. If the spacer is too short, the enzymes crash into each other. If it is too long, they cannot reach each other. Therefore, calculating the exact physical spacing is critical for the scissors to successfully cut the DNA. To ensure the enzymes can meet perfectly, we use the spacer distance formula. In this equation, L spacer is the length of the gap between the two talons where the actual cutting happens. The variable x2 is the starting position of the right talon on the DNA strand, and x1 is the starting position of the left talon. Finally, L binding represents the length of the DNA sequence that the left talon physically covers. By subtracting the starting position of the left talon and its binding length from the starting position of the right talon, we find the exact number of base pairs left empty in the middle. Let us put this spatial geometry into practice with an example. Suppose the left talon binds at position 100 on the DNA strand, and the right talon binds at position 135. We will assume the left talon covers exactly 20 base pairs. We need to determine if the spacing between them is optimal. We set up our equation and substitute our values. 135 minus 100 minus 20. This gives us a spacer length of exactly 15 base pairs. For the FOC1 enzyme to work properly, the spacer region must typically be between 12 and 20 base pairs long. Because 15 falls perfectly within this optimal range, the two domains will have just enough room to bind together and cut the DNA. To truly cement our understanding, let us look at this schematic diagram of the TALEN system in action. Here, you can see the double-stranded DNA helix running horizontally. On the left TALEN protein is bound to the top strand, reading its specific sequence. On the right TALEN is bound to the bottom strand. Notice the exposed stretch of DNA between them. That is the 15 base pair spacer region we just calculated. Attached to the inner ends of both talons are the circular FOC1 domains. You can clearly see how they reach across the spacer region to meet in the middle, forming a dimer. Right at that junction, the double-strand break occurs, executing the precise genetic edit. We have covered a lot of ground today, so let us summarize the key takeaways. First, TALENs are incredibly powerful tools that combine a customizable DNA reading domain with a strict DNA cutting domain. Second, because the cutting enzyme requires a partner to function, scientists can target unique sequences within billions of base pairs, preventing dangerous off-target mutations. Third, we saw how understanding the mathematics of sequence probability and the physical spacing required for these enzymes is the absolute key to mastering their design. You now understand both the biology and the math behind one of the most advanced genetic engineering tools in the world. Great job!